Hello, my name is Michael Kühl. I'm a professor for biochemistry at Ulm University. Here I'm the director of the Institute for Biochemistry and Molecular Biology, and I'm the chairman of the International Graduate School in Molecular Medicine Ulm. Our research focus lies on the characterization of intracellular signal transduction pathways. Today I would like to give you an introduction into intracellular signal transduction and its role in cancer. The major aim of this unit is to provide you with a refresher in basic principles of growth factor signaling and, of course, its relevance in tumor biology. So we start with a general introduction into the principles of signal transduction. Then we focus on some examples, for example, the role of tyrosine kinase receptors in signal transduction, or the protein RAS, which is a central player in many signal transduction pathways. We will talk about GTPases as molecular switches and on other signal transduction pathways like JAK-STAT signaling, heterotrimeric G proteins, Hedgehog signaling, or Wnt signaling. Note that growth factors are important because they regulate the cellular behavior. They can drive the cell cycle and thereby cell proliferation. They can trigger apoptosis, meaning cell death. They can regulate the cell migration, which is an important issue for metastasis, or they can regulate cell polarity. Note that in cancer cells, the signaling of growth factors is uncoupled from the biological response, meaning that cancer cells do not require an extracellular stimulus to divide. Cancer cells can bypass the requirement for growth factors, and cancer cells can generate their own mitogenic signal. Let's have a look on the principles of signal transduction. Growth factors are in general produced by cell A, are then released into the extracellular environment, the bloodstream, for example, and can bind to receptors on a different cell depicted as cell 2 here, and thereby activate signal transduction pathways in these cells. Thereby, receptors transduce the information from outside the cell into the cell. There are other examples how signal transduction can occur. Retinoic acid, for example, is a substance that is produced by a cell A, or 1, released, and then at the target cell can pass through the cell membrane and find an intracellular receptor and activate target gene expression. Another example is shown in panel C. The ligand, for example, for the receptor can also be presented on the cell surface of a neighboring cell. And finally, in D is shown that two cells can couple tightly together through gap junctions and thereby allow the flux of ions from one cell to another cell, thereby also allowing cells to communicate with each other. There is an enormous number of growth factors that can be categorized, however, into different families, like the fibroblast growth factor family, the transforming growth factor family, the epidermal growth factor family, and so on. Also, the receptors can be categorized, basically on their structure and on their functional properties. Most receptors, as highlighted on one slide before, span the membrane several times. The extracellular domain can bind the ligand, whereas the intracellular domain activates intracellular signal transduction. Based on structure and properties, we can distinguish different types of families. Tyrosine kinase receptors, for example, have a tyrosine kinase domain in their intracellular domain, meaning that this domain can phosphorylate tyrosine residues on target proteins. Similar, serine-threonine kinases can phosphorylate serine-threonine residues. In some cases, the receptor does not have a kinase domain, but can associate with intracellular kinases. Another important group are 7-transmembrane receptors that pass the membrane 7 times. Some ion channels are also regulated by extracellular ligands. And as shown for retinoic acid, some receptors are also found intracellular. So how does the ligand binding result in the activation of a receptor? The major issue here is that the binding of the ligand to the receptor results in a change in the three-dimensional structure of the receptor, as shown here. The biochemist would say that there is a conformational change. And this conformational change results, for example, in the intracellular activation of the enzymatic activity of the receptor, or the generation of binding sites for other proteins. And thereby the information is transferred from the ligand to the cytoplasm. Let's focus on this issue a little bit more in detail by focusing on tyrosine kinase receptors. Here, the binding of the ligand brings together two monomeric receptor units. The binding of the ligand activates the tyrosine kinase activity, which is found in the intracellular domain of the receptor. In a first step, these domains phosphorylate each other, which we call transphosphorylation. Intracellular tyrosine residues of the receptor are phosphorylated. By generating these phosphorylated tyrosine residues, docking sites for other proteins are generated. These docking sites are recognized by certain domains of the target proteins. For example, the so-called SH2 domain, because it's found in a protein which is called Src. So SH2 is the Src homology domain. With this SH2 domain, some proteins can recognize phosphotyrosine residues that are flanked by certain other amino acids. Here in this example, two adapter proteins like GRB2 and SOS link the receptor tyrosine kinase to a further downstream mediator, which is called RAS. So protein-protein interaction is an important issue for intracellular signal transduction. There are many binding domains known and have been identified that are involved in the recognition of certain properties. As seen on the last slide, the SH2 domain is able to interact with phosphotyrosine residues. The same holds true for the PTB domain. Other domains like the SH3 domain can interact with proline-rich regions in proteins. The 14-3-3 domain is able to recognize phosphoserine residues. The pH domain is able to identify phosphorylated inositides that are certain lipids. The bromo domain can identify acetylated lysines, or the C1 domain can identify or interact with diacylglycerol, a lipid in the membrane. A nice example how these domains are involved in intracellular signal transduction is a protein which is called BCR-ABL. This is a fusion protein that is generated by a chromosomal translocation. Normally, ABL is a tyrosine kinase involved in signal transduction. Through the fusion with BCR, this tyrosine kinase domain is deregulated, and its signal towards a deregulated proliferation for example in chronic myelogenous leukemia or CML. There are, however, multiple other domains in the BCR-ABL fusion protein that are able to activate different other signal transduction pathways, like the SH2 domain, the pH domain, or the GAP domain. Let's return to the tyrosine kinase receptors. There are many examples that changes in tyrosine kinase receptor signaling can lead to cancer or are at least involved in cancer. For example, mutations in the receptor can result in a ligand-independent activations. For example, mutations in KIT are found in gastrointestinal stromal tumors, or mutations in FLT3 that result in a length change or internal tandem duplication are involved in AML. Deletion of the active domain can result in an active receptor because it's not regulated anymore by the ligand. Mutations in the EGF receptor, for example, that fulfill this criterion are found in glioblastomas. Fusion with other proteins can also result in artificial dimerization and activation of the receptor, like RET in endocrine neoplasias. Overexpression of the receptor are found in multiple tumors, HER2 for example in breast adenocarcinomas. Also, overexpression of the ligand can result in a constitutively active pathway, for example the overexpression of PDGF in osteosarcomas. So let's have a first question in between. So which strategies could you envision to interfere with tyrosine kinase receptor signaling in cancer? So the answer is that you have to somehow interfere with the active receptor. And there are, for example, antibodies directed against tyrosine kinase receptors like Herceptin, or there are also inhibitors that target the intracellular kinase domain, such as Labatinib. So let's continue with the intracellular signal transduction. Remember that we talked about this mode, the protein RAS. RAS belongs to a family of proteins which are GTPases. These are in principle molecular switches because they occur in two different activity states. When loaded with GTP, for example, they are active. When loaded with GDP, they are inactive. Interestingly, GTPases have the ability to cleave GTP into GDP and phosphate. Thereby, they can switch from an active state into an inactive state on its own. If we remember RAS would be active in this GTP-bound state and would be inactive in this GDP-bound state. There are additional proteins that regulate this small circle here. So GTPase activating protein accelerate the cleavage of GTP into GDP and thereby turn off the GTPase RAS, whereas guanine nucleotide exchange factors facilitate the reloading of GTP and thereby activate the GTPase. So RAS belongs to this family of monomeric GTPases, and once activated through a tyrosine kinase receptor pathway, it is involved in different signaling pathways further downstream, like the PI3 kinase pathway, the RAF kinase, or the RAL-GEF. To fulfill these functions, RAS is farnesylated, meaning that it's lipid modified, and thereby it is attached to the cell membrane. This is an important issue because RAS needs to be lipid modified in order to be active. Now let's focus onto these different further downstream pathways. RAS, for example, can regulate gene expression by activating a kinase pathway downstream of it. This involves the protein RAF and other kinases that in a cascade transport the information towards the nucleus. At the end, a transcription factor is activated through phosphorylation, thereby allowing the transcription of target genes. There is a certain class of target genes which is called the immediate early genes. These are direct target genes of a signaling pathway, and they do not require the translation of other proteins in between. So the response is very rapid and occurs in less than 1 hour. Here, for example, it involves a protein like FOS, JUN, the serum response factor, or MERK. As mentioned before, RAS can also activate PI3 kinase. What's the role of PI3 kinase? PI3 kinase generates phosphatidylinositol 3,4,5-trisphosphate, also called PIP3, by phosphorylating a substrate diacylglycerol. PIP3 is a lipid and is recognized by other proteins like the protein kinase B, PKB, also called ACT, through its pH domain. ACT then can activate further downstream mediators, which we will discuss in a minute. Another mediator of PIP3 is ROGAF. So GAFs, remember, are guanine nucleotide exchange factors. So ROGAF is an activator of Rho, and we will also talk about this in one of the next slides. Importantly, the activity of PI3 kinase is counteracted by a phosphatase which is called PTEN. The PI3 kinase pathway is also important for cancer. This is because PKB has positive effects on cell proliferation, cell survival, and cell growth. And this occurs because PKB has anti-apoptotic effects through modulating the function of BAT, caspase-9, FOXO1, or MDM2. And at the same time, PKB supports cell proliferation by modulating GSK3-beta, FOXO4, and P21. In many tumors, PI3 kinase is hyperactive. In other tumors, mutations in PTEN are found. In both cases, the concentration of PIP3 is increased. So just as an example, mutations in PTEN are found in glioblastomas, breast carcinomas, or prostate cancers. And there are many other examples known. PIP3 also links to the mTOR pathway. The mTOR pathway is important because it regulates protein synthesis and therefore is required for cell growth, which is a prerequisite for cell division. So mTOR-RAPTOR is a complex of two proteins that facilitates protein synthesis and thereby cell growth. mTOR can also interact with a protein called RICTOR, thereby activating ACT-PKB and thereby supporting cell survival and cell proliferation. So mTOR is in a critical position to allow cell growth and cell division. Interestingly, there is an inhibitor called rapamycin that is an inhibitor of mTOR signaling. rapamycin can interact with a protein called FKBP12, which can inhibit mTOR function. And therefore, many mTOR inhibitors are already tested in clinical trials in different cancer types, for example, renal cell carcinomas. Turning back to RAS and its downstream signaling mediators. We already heard that RAS can activate different guanine nucleotide exchange factors, GAFs. The important thing with these proteins is that at the end they can regulate road GTPases like CDC42 or RAG. And these are proteins that are required for cell migration. Migration of cells is characterized by the formation of cellular extrusions called lamellopodia or filopodia. And both of these extrusions are required for cell migration and therefore are required for metastasis. They do so by regulating the actin cytoskeleton. As such, GTPases can be turned on indirectly by activated RAS, as shown for one example here on this cartoon. RAS can regulate cell migration. Let's think of the RAS protein in cancer. So RAS is in a central position in many signaling pathways. Can you envision how a drug could interfere with RAS function? Remember that RAS needs to be lipid modified to be attached to the membrane and to function properly. So RAS farnesyl transferase inhibitors that interfere with this lipid modification can block the function of RAS. So in the following, we would like to concentrate on another signal transduction pathway, which is activated by transforming growth factor beta growth factors that activate serine threonine kinase receptors. There are many members of this growth factor family, and the main characteristic of this pathway is that the binding of the ligand results in dimerization of two different receptor types, 1 and 2, as shown here on the cartoon for BMP. Binding of the ligand results in the activation of a kinase activity in the intracellular domain of the receptor and in receptor phosphorylation. These phosphorylated residues are now binding sites for other proteins, which are called SMAD proteins. There are different types of SMAD proteins. And the so-called R-SMADs are those SMADs that can bind to this phosphorylated residue on the receptor, thereby are phosphorylated itself, and then can transduce the signal inside the cell. Phosphorylated receptor SMADs interact with so-called co-SMADs, for example SMAD4. This complex can enter the nucleus and can act as a transcription factor. There are many pancreatic and colon cancers that have inactivated SMAD4 proteins. An explanation might be that TGF-beta signaling interferes with c-Myc action. Another example for intracellular signal transduction are receptors that activate JAK-STAT signaling. Many cytokines can activate this pathway. And receptors for these cytokines do not have an intrinsic kinase activity. Instead, they can interact with intracellular kinases like JAK. These kinases are activated by the binding of the ligand. These activated kinases then can transphosphorylate either the kinase or the receptor. Thereby, docking sites are generated for other proteins called STAT. These STAT proteins are then phosphorylated by the activated JAK, form a dimer, enter the nucleus, and activate target gene expression. In many breast cancers, for example, STAT3 is constitutively active. And there are also examples known where mutations in JAK play a role, for example, in myeloproliferative disorders like polycythemia vera or essential thrombocythemia. Another class of receptors are 7-transmembrane receptors. They span the cell membrane 7 times and in most cases are coupled to so-called heterotrimeric G proteins. These heterotrimeric G proteins have 3 subunits. The G-alpha subunit has a GTPase activity. So it can bind GDP and is active, and it can bind GTP and is active. So what happens if a ligand binds to the receptor? The 7-transmembrane receptor undergoes a conformational change and thereby facilitating the release of GDP and the binding of GTP. In this way, the ligand-activated receptor behaves like a guanine nucleotide exchange factor for the G-alpha subunit. Furthermore, the heterotrimeric G protein dissociates into an active alpha subunit and a beta gamma subunit. Both the G-alpha subunit and the G-beta gamma subunit can activate intracellular signaling. Just 2 examples are shown here on this slide. An activated G-alpha subunit, for example, can activate the phospholipase C-beta, thereby generating second messengers like cAMP or IP3, and thereby activating calcium signaling, for example. Also, the G-beta gamma subunit can activate downstream mediators, such as phospholipase C-beta. G-protein coupled receptors are also involved in cancer. Just to give you some examples, a constitutive active mutation in G-alpha S can be found in thyroid cancers or pituitary cancers. Mutations in a receptor can also lead to a constitutive active receptor. And examples are the TSH receptor, FSH receptor, or LH receptor. And interestingly, some viruses encode G-protein coupled receptors that are also involved in tumors, for example, Kaposi sarcoma. Let's now focus on Hedgehog signaling. The receptor for Hedgehog is a protein which is called patched. The main property of patched is to inhibit another transmembrane protein which is called smoothened. In the absence of Hedgehog, patched is inhibiting smoothened. If, however, Hedgehog binds to the receptor, this inhibition is released, so smoothened now is active. Now let's turn to the intracellular part of the pathway. In the absence of Hedgehog, a transcription factor, which is called CI or GLI, is processed into a transcriptional repressor. If, however, the pathway is active, GLI acts as an activator of transcription. Hedgehog signaling can also be involved in cancer. There are mutations known in patched and smoothened that have been found in basal cell carcinomas of the skin, or mutations in patch that have been found in medulloblastomas. So which properties must have a substance that should interfere with the constitutively active Hedgehog signaling in cancer? Remember, mutations in patched or smoothened activate the pathway. Such a substance should therefore interfere at this level or further downstream. Such a drug, for example, is cyclopamine, which is found in the corn lily. Cyclopamine inhibits the activated smoothened receptor. Cyclopamine is therefore currently being investigated as a treatment agent in several tumors like basal cell carcinoma, medulloblastoma, rhabdomyosarcoma, glioblastoma, and multiple myeloma. The last pathway that we would like to discuss today is the Wnt signaling pathway. Wnt proteins are secreted glycoproteins. This is a large family of growth factors. 19 members are known in mammals. To activate intracellular signal transduction, they bind to receptors of the frizzled family and several co-receptors such as LRP5 or 6. Based on the high number of ligands and also receptors, it's not surprising that Wnt proteins can activate different intracellular signaling pathways. Wnt proteins can regulate the cell cycle, cellular differentiation, cell polarity, and migration. The best characterized pathway is the canonical Wnt pathway. The major player here is a protein which is called beta-catenin. In the absence of a Wnt ligand, cytoplasmic beta-catenin is degraded by the proteasome. For degradation, beta-catenin is bound to a multi-protein complex, which is called the destruction complex, consisting of 3 proteins called APC, axin, and GSK3-beta. Once beta-catenin is bound to this destruction complex, it is phosphorylated by a dual kinase mechanism at certain serine and threonine residues. This phosphorylation targets beta-catenin for modification by ubiquitin and subsequent degradation by the proteasome. So, as a consequence, in the absence of a Wnt ligand, the cytoplasmic level of beta-catenin are low. In presence of a Wnt ligand, the destruction complex is disassembled. As a consequence, beta-catenin is not phosphorylated and it's not targeted to the proteasome. Concentrations of cytoplasmic beta-catenin are increasing, and subsequently cytoplasmic beta-catenin can interact the nucleus. In the nucleus, beta-catenin interacts with transcription factors of the TCF-LEF family, where TCF stands for T-cell factor and LEF stands for lymphoid enhancer factor. So these proteins were first described in T and B cell development. Target genes of this complex include those required for cell cycle regulation, such as cyclin D1 or c-Myc, and by these target genes, the canonical Wnt beta-catenin pathway is linked to a positive regulation of the cell cycle. Let's have a look onto this cartoon to summarize the Wnt pathway. So the major player is beta-catenin, which can be phosphorylated for degradation. This phosphorylation is facilitated by the destruction complex consisting of GSK, axin, and APC. If, however, a Wnt ligand is present, the destruction complex disassembles. Beta-catenin concentrations can increase, go into the nucleus to interact with TCF-left transcription factors to activate target gene expression. There are mutations known that link this pathway to cancer formation. There are, for example, mutations known in APC that are involved in colon cancer. What does APC stand for? It means adenomatoous polyposis coli. If we have a look onto the structure of APC, we can see that there are different regions in the APC protein that are required for beta-catenin binding and degradation. Found mutations in APC result in a truncated version of the molecule. Interestingly, there are hotspots of mutations that lead to a truncated version of the molecule here. And if you have a look, this is the region where beta-catenin needs to bind and which is required for subsequent beta-catenin degradation. So these mutations of APC result in an increase in cytoplasmic beta-catenin. The pathway is constitutively active. Thus, APC is a tumor suppressor gene. Note that we will have an extra lecture on tumor suppressor genes. Also, mutations in beta-catenin can result in pathway activation. As we have heard, beta-catenin is phosphorylated at different sites. The critical residue here is serine 33, which is required for subsequent degradation. Mutations affecting serine 33, for example by a point mutation or by deletion of several amino acids in this region, therefore result in a stabilized version of beta-catenin which cannot be targeted to the proteasome anymore. As a consequence, Wnt beta-catenin signaling is constitutively active. As we have heard, there are several pathways that can be acted by Wnt proteins. So far we focused on the beta-catenin pathway. Pathways that do not involve beta-catenin are called by definition non-canonical Wnt pathways. And there are basically two that we should discuss. The Wnt calcium pathway involves the intracellular release of calcium ions, whereas the Wnt JNK signaling pathway involves activation of Rho GTPases and Jun N-terminal kinase JNK. Interestingly, non-canonical Wnt signaling can inhibit the canonical Wnt beta-catenin pathway, so Wnt ligands that can activate non-canonical Wnt signaling can therefore function as tumor suppressor genes. Let's also have a look onto this cartoon. On the left hand you see the Wnt JNK pathway that involves Rho or RAC to activate Jun N-terminal kinase and to regulate the cytoskeleton, for example. On the other hand, the Wnt calcium pathway involves the release of intracellular calcium, the activation of further downstream calcium-sensitive enzymes like calcium calmodulin-dependent kinase 2, protein kinase C, or the calcium-sensitive phosphatase calcineurin. Non-canonical Wnt signaling has different functions in cancers. The first is, as we have heard, non-canonical Wnt signaling can inhibit canonical signaling. So Wnt5a has been shown to have a tumor suppressor activity in different tumors like thyroid carcinoma. Wnt5a also inhibits B-cell proliferation. On the other hand, non-canonical Wnt signaling regulates the actin cytoskeleton. And as we have heard earlier in this lecture, regulation of the cytoskeleton is a prerequisite for cell migration as we have it in formation of metastasis. Wnt5a signaling enhances motility in melanoma cells. And the pro-invasive activity of Wnt, for example, has also been shown to be mediated through a non-canonical Wnt pathway. So if we think of Wnt signaling, which proteins would you consider to be druggable targets in a patient having a colon cancer where mutations in APC were found? So the drug has to act on proteins downstream of the stabilized beta-catenin. So we would have to look for a substance that interferes with the interaction of beta-catenin with TCF-left transcription factors, and therefore beta-catenin or TCF-left could be targets for this substance. So we are coming to the end of this lecture. As you have seen, understanding the molecular mechanisms of intracellular signal transduction provides us with insights how a cell regulates cell proliferation under physiological conditions. Misregulation of signal transduction pathways can lead to growth factor independent repetitive rounds of cell division. Understanding misregulated signal transduction in cancer provides us with hints how to develop anti-cancer therapies. And basic principles and examples for these issues were discussed in this lecture. So, thank you very much for your attention. I hope I was able to give you some insights into the basic principles of intracellular signal transduction and its role in cancer. I hope you enjoyed this lecture.